Good afternoon to everyone. I would like to discuss with you today the mechanism of action of three classes of drugs, all belonging to the reverse transcriptase inhibitors that still remain the cornerstone of the therapy of HIV, even 30 and more years after the beginning of antiviral therapy. You see that we are highly concentrated on reverse transcriptase when we think about how to produce an effective antiviral regimen. Now we have a new class that is NRTTI, you see on the left of the slide, that we will discuss in detail in the next slides. And the molecular formulas of this class is very similar each other. You see they are all deoxynucleoside analogs modified mostly at the level of the pentose of the deoxynucleoside. These let the drug be recognized as a deoxynucleotide by the reverse transcriptase being incorporated into the DNA chain information. How do these kind of drugs work, particularly the NRTI, the nucleoside reverse transcriptase inhibitor, which is their mode of action? Their mode of action is relatively simple. There is a continuous translocation of a new nucleoside, deoxynucleosides, triphosphorylated, that thanks to the reverse transcriptase are added to the DNA chain information. In this way, the genomic RNA above is replicated in a complementary sequence of viral DNA that becomes at the end a double-strand DNA, thanks again to another function of the reverse transcriptase. So what do the NRTI do? The NRTI are very similar to the classical deoxynucleosides. They are recognized by the reverse transcriptase and introduced like they are the normal deoxynucleosides in the DNA chain information. Where is, however, the difference? The difference is that this drug does not contain usually a particular structure at the level of the pentose that does not let the next deoxynucleoside be added in the DNA chain information. So in other words, what you can see is that while the translocation still occurs because new drugs, new deoxynucleotides are put close to the reverse transcriptase, there is no chance to add it into the DNA chain information. That is the reason they are called DNA chain termination. Which is the mechanism of action by which the major drugs work, essentially. As we said, they act by blocking the reverse transcriptase and block the DNA chain structure. So they are called, as I said, the DNA chain terminators. How does the virus react? The virus reacts by adding and modifying the structure of the reverse transcriptase at certain amino acid levels, peculiar amino acid levels. Here you see three examples: the M184V, typical mutation for 3TC and FTC resistance; the Q151M, that is a typical mutation code for multi-drug resistance; and the K65R, that is a mutation that is that confers resistance mostly to tenofovir, but also to abacavir. How does it work? You see that in red, you have the nucleoside analog, that is the drug that blocks the reverse transcriptase. But thanks to the mutation that the virus generates within the reverse transcriptase, the purple triangle, the nucleoside analog is no longer able to reach the reverse transcriptase. This is a classical method of resistance for tenofovir, lamivudine, and emtricitabine, as an example. Then we have other drugs like the thymidine analogs that instead produce a reaction by the virus different than the previous one. In this case, there is no modification of the reverse transcriptase in such a way that the nucleoside analog is no longer able to reach the reverse transcriptase. The reverse transcriptase is joined by the, for example, the AZT triphosphate. However, thanks to the consume of ATP molecule, it is possible to excise the nucleoside analog incorporated in such a way, you see below, that it is removed and the reverse transcriptase starts again its normal job. So you see in the previous slide and in this slide two different mechanisms by which the virus defends itself against the nucleoside analogs. Both of them are very effective. That is the reason that if the therapy is not given properly, the virus reacts by mutating and the drugs are no longer effective. Remembering that for many NRTI, there is a cross-resistance. That is that, as you can see, for example, in this slide, these mutations that you see listed here can produce a cross-resistance to zidovudine, AZT, and stavudine, D4T, but pulled together, they can produce a certain level of resistance also to other drugs of the same class, NRTI. Indeed, as you can see in this slide, while in a wild-type virus that is without resistant mutations, more or less all the drugs, all the nucleoside reverse transcriptase inhibitors act at the same, at certain level that we call zero, so a different potency, but all of them are active against HIV. If mutations start to appear, according to the different type of mutations, the drug reacts in different way in terms of efficacy. You can see that lamivudine and tricitabine lose practically all their efficacy in the presence of 184V or I mutation. Abacavir loses something, while, please note carefully, tenofovir, zidovudine, and stavudine become a little bit more effective. In other words, certain mutations might produce a full resistance. The same mutation against other drugs might produce increased susceptibility. This is one of the bases of that regulates the concept of multi-drug therapy, combination of drugs that even in the case the virus induces a mutation that make it resistant to one of the drugs, there is another drug that remains fully effective or even more effective thanks to the presence of that mutation. In addition, certain mutations in the reverse transcriptase caused by NRTI pressure might affect the fitness of the virus because they act like the 184V directly within the core of the enzyme in such a way that the virus, even if it escapes the capacity of the drug of blocking its efficacy, still it loses the capacity of replicating. This is what we call loss of fitness, that in terms of clinics has a substantial relevance, because we see that there are patients that even if carrying a virus resistant, even in this case, the virus does not proceed toward the high viral load. CD4 loss is relatively low, and the clinical conditions remain substantially stable, at least for a certain period of time. So resistance, fitness are two concepts that need always to be taken into account together in order to understand how the drugs work. And what happens instead if we have many mutations? This is a disaster. If you have, for example, the 184V plus 3 times the mutations for AZT, D14, etc., pulled together, we see that unfortunately, we can lose the entire class of drugs. That is the reason that we need to avoid strongly that these mutations appear. If these mutations appear, we need to use different drugs, because NRTI under these conditions do not work anymore. Let me go to NRTTI. A small difference in the name, a substantial difference in the mode of action. Islatravir has a dual mechanism of action. One of them is the classical NRTI, like NRTI inhibition, that is a chain termination, and we will see about that. But due to the peculiar structure of the molecule, you see that we have a group here, this 18 group, that causes a perturbation of the structure of the DNA chain, you see here, in such a way that not only we have a DNA chain termination, but we have also the inhibition of the translocation. In other words, there is no chance for the reverse transcriptase to bring closer to the remaining part of the DNA chain the next nucleoside. So it's a dual mechanism that let us the drug being very effective and with a high genetic barrier. We will certainly have the chance in other conditions, in other situations, to discuss more the issue of the genetic barrier. So we have a translocation inhibitor, you can see on the left panel, and we have a delayed chain termination that even if translocation occurs under the pressure of islatravir, still you have the chain termination. Therefore, this dual mechanism of action allows this kind of drug to be really potent, even at very, very low drug concentration. So this is the structure of the molecule. You see that it is an adenine derivative, highly modified at the sugar level, and each of the parts of the molecule plays a role in the capacity of the drug of being very potent and escaping the capacity of the virus to generate mutations that may reduce the efficacy of the drug per se. How is islatravir active within the cells? As every nucleoside analog, it is triphosphorylated in the triphosphorylated islatravir form that within the nucleus, it enters within the DNA chain, and the DNA chain is blocked, as we said before, which is the other point that makes this drug very interesting. Essentially, the fact that due to the characteristics of the drug, the triphosphate form has a steady state concentration within the cells, very high and very long. You see that can remain within the cells up to 191 hours. That is very, very long. This makes this drug suitable not only for the classical daily therapy that is provided to our patients, but also for the long-term therapy that is becoming more and more one of the issues of the new therapeutic approaches to HIV infection. In other words, there is a possibility that this drug is not administered every day, but can be administered every week or even once a month, or perhaps even more, because the concentration of drugs within the cells remains so high that maintain an efficacy against the virus, even if the drug has been administered a long time before. How is it translating in a virological concept? You can see very clearly from these two panels. On the left, you see that the efficacy of islatravir is 2 logs or even more greater than other drugs like tenofovir, like AZT, like tritisin, etc. So it has a potency very strong, even at very low concentration. And in the right panel, you can see that islatravir is very potent even against virus strains carrying mutations conferring resistance to the other drugs. Islatravir remains effective also in the majority, against the majority of the mutated and resistant viruses. In other words, this drug can be used at the beginning of the therapy, but can represent a very important rescue in those patients that, being highly resistant to NRTI, no longer can afford to be treated with this kind of drugs. There are some evidences that also against islatravir, the virus is able to produce some resistance, but you can see that the resistance is quite limited, not very effective, and pretty often different than what is found with the NRTI. In other words, completing the journey on islatravir, we have a drug that has a high genetic barrier that is able to interact very strongly with the reverse transcriptase of the HIV, and therefore is very promising for the clinical practice, both for the first line of therapy, for the last lines of therapy after resistance, but also for as a long-acting drug that can be administered pretty rarely over time. What can we say about NNRTI? Far less, because this class is less complicated than the class of NRTI and NRTTI. We can say, however, that the original NNRTI were drugs very similar, nevirapine, delavirdine, and efavirenz, and relatively similar were etravirin and rilpivirine, the second generation of this class, that act with the same mechanism against the reverse transcriptase, by blocking not the active side, but blocking the flexibility of the enzyme that at that point is no longer able to accept the new nucleoside coming from within the cytoplasm of the cells. So you see that there is no space anymore, thanks to the drug, to accommodate a new triphosphate nucleosides. This is very good, except the fact that the virus is very capable of escaping. Even single mutations let the virus escape from these kind of drugs. Very potent, but very low genetic barrier. And the resistance of the 4 or 5 different drugs that have been used in clinical practice is relatively similar. That is, if one of the drugs of this class is lost, the whole class usually is lost, with the minor exceptions. In other words, NNRTI need to be used with the caution and usually at the beginning of the story of our patients. The situation is not the same for doravirine, because doravirine instead is a drug with a different chemical structure that has the peculiarity of being effective also against resistant viruses carrying mutations that confer resistance to the first and the second generation NNRTI. In other words, is a new drug that makes new activity. You can see that the mutations that occur during the therapy with doravirine are different than the classical one, but more important, that doravirine remains active even in the presence of the classical mutation that occur when the virus has become resistant against the classical NRTI. How to use it in clinical practice? First, the resistant associated mutation of doravirine are rare that they occur in clinical practice. There are some of our papers, but there are also papers by French colleagues and others that show the same result. What I feel really important, beyond the good activity of doravirine that makes a difference and suggests that this drug can be used alone also in patients that carry resistance to the classical NNRTI, I think the combination of doravirine and islatravir is the one that deserves attention in my last 2 minutes of presentation. Essentially, the mutations generated by doravirine and those generated by islatravir seem to have a counter effect in terms of resistance. In other words, the mutations that are produced by doravirine, like the 227C, and the mutation induced by islatravir, that can be the 184V, the two seem to increase the efficacy of the other drug. That means in practice that a virus put under the pressure of the two drugs together doesn't know where to go, because if it goes toward the resistance to doravirine, makes more effective islatravir and vice versa. This is a classical combination in which there is always a very, very good convenience in using the drugs together. So the cell culture selection that has been showed by the group of Bluma-Bremer clearly show, as you can see here, the first group in which doravirine is used alone, in which there are many mutations. When doravirine is combined with islatravir, the number of mutations, the type of mutation is reduced, as it is the case of doravirine plus TTC that causes the same mutations of islatravir. So these are the basis of the concept of using these drugs together, and this is something that certainly it is done in the future. So going toward conclusions, I think we need to think new in our patients. We have new drugs, we have to use them properly. We have to consider that the cross-resistance amongst the drug that we're using today is very limited. And so the rational approach requires still the use of reverse transcriptase inhibitors, but with a rational abuse. Thanks to everyone for the attention. So.